Hello, good morning. Welcome to our first technical session. In this session, we will have 4 presentations, 2 full papers and 2 short papers. And the audience that was watching us on YouTube, you can make questions concerning any of those presentations. So please write down your questions in the comments box in our YouTube channel. And the questions, they will be directed to the speakers after the last session. So we will have our presentations in a straight way, the 4 of them, and after the presentations, we will deliver the questions to the speakers. So uh, now I'd like to introduce our first speaker. Is Fernanda Demarchi. She will address us the talk entitled Hyposine plays a role in the translation of short mRNAs and mediates the polyamine and autophagy pathways and Saccharomyces cerevisiae. So, Fernanda, you're gonna present a full paper. You have 20 minutes sharp to deliver your talk. Thank you so much for your.
Thank you, Marcelo. Good morning, everyone. So we will start.
Hello, everyone. My name is Fernanda. I'm a Master in Biotechnology and a PhD candidate in Sciences. And today I'll present a paper entitled Hyposine Plays a Role and Mediates the Polyamine and Autophagy Pathways in Saccharomyces cerevisiae. The first author of this paper is Ana Carolina Paiva, and I'm one of the authors. So first of all, I'd like to remember the protein synthesis process with you. Translation is the process where ribosomes and translational factors recognize and decode the messenger RNA template by cycling through translation initiation, elongation, termination, and ribosome recycling phases. In the initiation in eukaryotes, the 40S and the 60S subunity, ribosomal subunities, make up the 80S ribosome, which travels through 3 binding sites: the aminoacyl, the peptidyl, and the exit sites. At the start of translation, initiation factors called EIFs in eukaryotes drive the proper assembly of the ADS ribosome at the start codon with the initiator tRNA linked to the P site. Moving on to translation elongation, in which the synthesis of the polypeptide chain occurs, coordinated by elongation factors that facilitate the binding of aminoacyl tRNAs at the A site in a codon-dependent manner here, while the tRNA at the A site is translocated to the P site, forming a hybrid state in relation to the ribosome subunities. and with the concomitant ribosome translocation by mRNA. Translation is completed when the ribosome encounters a stop codon and releasing factors induce the hydrolysis of the polypeptide. And finally, the ribosome recycling, which induces the separation of subunits for a subsequent translation process. The protein that we will focus on this work is related to the translation elongation and is an elongation factor. It's called EIF5A. So some of you may think, oh, but isn't this an initiation factor? But there is an explanation. EIF5A was first described as a initiation factor, but nowadays it's well established and described that EIF5A works actually during elongation of during translation elongate. So EIF5A undergoes hypogenation, that is an exclusive and spermidine-dependent post-translational modification that converts a specific lysine residue by the action of two enzymes, deoxyribosyne synthase and deoxyribosyne hydroxylase. This in yeast, okay, in Saccharomyces cerevisiae. Like the EIF5A, these enzymes involved in this exclusive modification are also evolutionarily conserved. EIF5A is essential for cell viability in eukaryotic cells. Hypogenated EIF5A promotes the stabilization of the peptide bond during the elongation of a subset of mRNAs with lower translational processability. Spermidine is a polyamine and hypogenation is a process that is spermidine dependent. And besides being involved in hypogenation, spermidine has also been reported to induce autophagy. Additionally, polyamines have roles in cellular proliferation, binding DNA, modulating ion channels and protein synthesis, and modulating cellular protein synthesis. A structural analog of spermidine is DC7 that acts as an inhibitor of the first hypogenation enzyme, anchoring at the spermidine binding site of this one. So, for the involvement of EIF5A and DC7 in autophagy is not clear by now. Their roles have been postulated as really controversial. For example, Patel and collaborators in 2009 described that the deoxyribosyne hydroxylated mutants and EAF depletion have increased autophagy, while for Oliverio and collaborators from 2014, EIF5A depletion had no interference in autophagy, while DC7 treatment increased autophagy. For Lubas and collaborators in 2018, EIF5A was required for autophagy induction. So to continue, let's understand what autophagy is. This is a catabolic process of lysosomal or vacuolar degradation, essential for promoting the recycling of cytoplasmic components and damaged or unnecessary constituents, maintaining cell homeostasis. In Saccharomyces cerevisiae, autophagy starts close to the vacuole, here being the site of assembly of the pre-autophagosomal structure. The initial phases consists of the formation of double-layer membrane structures called phagophores, which sequester intracellular components to form a closed spherical structure, the autophagosome, which fuses with the vacuole for acid hydrolysis of the captured charge, degradation, and subsequent efflux of nutrients to the cytoplasm. These structures are formed by proteins called ATGs. This first group of proteins form a ternary complex which associate with the phagophore, helping in its formation and elongation, until the formation of the autophagosome is in which they dissociate. The second group performs the conjugation of ATG8 with phosphatidylethanolamine, which associates with the membrane and helps to close the phagophore, forming the autophagosome and also aids in fusion with the vacuole. So the aim of this project, of this work, was to answer the question, what effects do EIF5A and DC7 have on autophagy? To answer this question, we used data sets of transcriptional and translational profile obtained by the Markian collaborators in 2020, a work presented in this same symposium last year by me. So we have data for transcriptional and translational profiles of a GIS11 mutant, a mutant of the oxyhypocene synthase, the first enzyme from hyposination. And these data are from the RNA-seq of total mRNA of a cell and from the polysome profile, where only the polysomes were sequenced. For data analysis, we used TrimGalore to trim out adapter sequences and sequence reads with low quality. Bowtie was used to exclude non-coding RNA reads. Also, HiSat was used to align our reads to the genome sequence, to the genome reference file. And then annotate-to-seq was used for differentially expressed analysis. So we defined translational efficiency as the rate between the transcript abundance in the translatome to the abundance of the respective transcript in the transcriptome profiling. Changes in translational efficiency was calculated by annotate-to-seq using analysis of partial variance. Gene ontology terms and biosynthetic pathways of differentially expressed genes were determined using Eastmine database. So, as a result, we have that hyposination interferes with the gene expression of short ORFs, such as mitochondrial ribosomal proteins. We found significantly different ORF length distribution for up and down regulated mRNAs in GIS11 translational profile compared to the wild type, implying that ORFs recruited for translation in the mutant strain are on average longer than those for the wild type. While the translation of short ORFs was strongly affected, these results were not observed for the transcriptional profile. It's already established that short ORFs are the most highly expressive ones, encoding small proteins that tend to play important maintenance roles in the cell, and their translation are related to the EASC1 protein, a component of the 40S ribosomal subunit. As observed in ASU1 mutant, ORFs under 500 nucleotide in GIS11 mutant presented lower translation efficiency values. Additionally, GIS11 mutation is synthetically lethal when combined with the ASU1 mutation, and the overexpression of GIS1 gene is toxic to an ASU1 mutant strain. In the present study, ASU1 gene is downregulated in the translational profile. Short mRNAs that code for mitochondrial ribosomal proteins are sensitive to the loss of ASC1 protein. So also, YDJ1, a putative cytosolic factor involved in mitochondrial protein import, seems to have the total mRNA levels downregulated in GIS11 mutant. These results were obtained by qPCR. When it comes to the polyamine pathway, Most of the genes and their respective transcriptional factors have changed transcriptional and translational levels in the GIS1 mutant. These results suggest that GIS11 mutant generate an imbalance of polyamines levels. Those polyamines are essential for many basic cellular functions. Their intracellular levels are tightly regulated in their biosynthesis, catabolism, and/or transport. So polyamine intracellular imbalance could affect some cellular processes, such as transcriptional, translation, gene expression regulation, stress resistance, and autophagy. In fact, we observed that most proteins involved in autophagy process have an increased translational efficiency in GIS11 when compared to GIS1, configuring an increase in these mRNAs recruited for translation. Additionally, Western blot analysis of two ATGs in wild-type strain with loss of hypoxenation caused by treatment with GC7 showed an increase in the protein level for both proteins. The ATG1 protein acts in the regulation and signaling to induce the formation of autophagosomes, and ATG33 is a specific mitophagy protein. GC7 treatment also attenuated anoxia-induced generation of reactive oxygen species in the cells and its normoxic conditions and increased the mitochondrial oxygen consumption rate of cultured cells in mice. Based on the above considerations, polyamines and DIA5A can modulate autophagy, and hypoxenation could be the key of this. So finally, our data suggests that hypoxenation supports the expression of a set of short proteins, providing a possible explanation as to why lack of hypoxenation results in the cell respiration defect. This is a consequence of translational problems due to the lack of translational processivity, as well as to the cellular imbalance of polyamines, resulting in long arcs occupying ribosomes. As a response, to maintain cellular homeostasis, there is an induction of macroautophagy and mitophagy. So thank you everyone for listening to me. If you have any questions, I'll be here to answer all of them. And you can find me at this email address right here. Thank you.
Thank you so much, Fernanda, for your nice presentation. So, as we said previously, we're gonna move for the next presentation, and the questions for your talk we're gonna deliver in the end of this technical session. So the next speaker is Pamela Miranda. She presents the work entitled Mesoscopic Evaluation of DNA Mismatches in PCR Primer Target Hybridization to Detect SARS-CoV-2 Variants of Concern. Hello, good morning, Pamela.
Good morning, Marcelo. Thank you.
So your presentation is a short paper presentation, so you have 10 minutes to deliver your talk.
Okay, thank you. Just share. Okay. Good morning, everyone. I'm Pamela, and I will start about my work with PCR, or polymerase chain reaction. It is a molecular technique used in several situations, and one of its elements is the primers. Usually a pair of primers called forward and reverse, or sense and unsense. So here we have an example of a pair of primers from our work. And they are a short DNA sequence. PCR has three main steps. The first one is the denaturation, in which the double strands are dissociated into single strands. The second step is the annealing, in which the primers, here in red, hybridized to the target or template. And the third step is the extension, in which the DNA polymerase amplifies the product. These steps are repeated usually from 30 to 50 cycles, and each one is carried out in a different temperature. In our work, we focus in the second step, uh, uh, when the primers hybridized to the target, in which the presence of mismatches may impact the reaction. But okay, what are mismatches? It is known the canonical base pairs, C and CG, which form the DNA sequence. So, in opposite of it, mismatches are incorrect pairings. So, one base binds to a non-counterpart. And there are 80 single mismatches. Mismatches are usually associated to the destabilization of the molecule structure, changing the conformation of hydrogen bonds and stacking directions. Here you can see an example, GA mismatched pair in red and GC canonical base pair in gray. So we can observe a slight difference in the conformation between both. Also, mismatches affect the melting temperature, which is the parameter we focus in our work. However, some types of mismatches are more stable than others, even the AT canonical base pair. And regarding our work about primers, when we have primers with a few mismatches, they may contribute to the stabilar hybridization in comparison to primers with no mismatches and with rich innate base pairs. One element which affects the behavior of mismatches is their neighborhood. So the base pairs which flank the mismatch base pair on both sides is relevant for study them. And there is 3 types of neighborhood. One of them is when we have the mismatch flanked on both sides by canonical base pairs. The other one is when we have one mismatch base pair and a canonical base pair. And the last one is when we have a triple mismatch. So one mismatch base pair flanked on both sides by mismatch base pairs. It is interesting, in the first 2 examples, we have the same primer hybridized through the sequence. And for a single mismatch, we have 52 degrees for melting temperature. Although we expect an increase in the melting temperature when we consider double mismatch for the same primer, it is the opposite. The melting temperature increases to 55.5 degrees. So it is very interesting. However, this calculation, it is not a simple rule, and it's necessary more complete calculations. One origin of mismatch is mutations. So mutations in the genome may lead to mismatch. It is the case of SARS-CoV-2. So we have a hybridization between prime and target of original genome. All these examples is the same primer, but when we consider hybridization through the 4 variants of consent of SARS-CoV-2, we can see many mismatches. So in some hybridizations, it is mainly mismatch, and others only one, for example. So from this, our question is, are these PCR primers able to detect SARS-CoV-2 variants of concern and not fail the diagnostic tests? To investigate it, we collected 21 primers and probes from hook protocols um, designed in the early pandemic and genomes of variants of concern that have been classified so far. The first step of our workflow is align all primers against all genomes using the Smith-Waterman algorithm and considering two strand configurations. One is the genome sequence, like in FASTA file, and the other one, the complementary genome sequence. The first step is the calculation of hybridization temperature of all primes and probes, which we call the reference temperature, and the melting temperature of matched alignments, that is only canonical base pairs, and the mismatched alignments, considering up to 3 consecutive mismatches. For this calculation for mismatched alignments, we used a recently available thermodynamic parameter set for DNA mismatches. And the third and last step of our workflow is the calculation of coverage for matched alignments and mismatched alignments. Here, we consider a limit. It is regarded as follows. We have the difference between the melting temperature of the primer and the melting temperature of the mismatched alignment of this primer. If this difference is equal or below the limit, so this alignment is accepted to the calculation and primer 2. So from our workflow, what we find? We find all primes from hook protocols achieved coverages above 90%, and most of them achieved at least 99% when we consider up to 3 consecutive mismatches. I brought up an example of the probe from Institute Pasteur. When we look to the coverage for matched alignments regarding to alpha variant, we achieved 99.7%. When we look for the same coverage, but in regards to delta variant, the coverage drops slightly to 98.9%. Remember, for both genome sets is about 7,000 genomes. So now, when we look for the mismatched alignments, the delta variant alignments increase to 100%. It is 84 more alignments, about 1% of all genomes of delta variants that we collected. So we can answer our question. And so when we consider the mismatches, the coverage increases for all variants of concern, alpha, beta, gamma, and delta, but especially for delta variant, we have a few cases of 100%. So from this, we suggest these primers should continue to be effective to detect SARS-CoV-2 variants of concern. Our flow is available in our webpage, so feel free to use it or contact us. Thank you to CAPS and CNPq. Thank you, everybody. And if you want to contact me, feel free to send me a message. Thank you.
Thank you so much, Pamela, for your presentation.
Thank you.
We address your questions at the end of the session. So next is presentation will be a recorded one. Unfortunately, the speaker could not attend here this session. We will present Mariana Santoni's presentation entitled Comparative Transcriptome Profiling of Meitinus ilicifolia root and leaf.
Hello, good afternoon. My name is Mariana, and I'm here to talking about my work. And my work is the comparative transcriptome profiling of Mytenius licifolia root and leaf. And this project was developed in São Paulo State University. And here are the authors of this work. So I am Mariana, and if you have doubts or if you need to talk to me, you can find me at my email address. So I'm going to introducing my work with talking about secondary metabolism. So different from animals that use movement to interact with the environment, plants respond to stimulus by producing compounds. The so-called secondary metabolites are substances involved in plant defense, communication, and response to stress. The metabolism in plants can be categorized into primary and secondary metabolites. Primary metabolites such as carbohydrates, lipids, and proteins are involved in plant development and cell growth. In contrast, secondary metabolites are multifunctional metabolites produced as an evolutionary adaptation. So they are different. And mainly secondary metabolites are small molecule. Secondary metabolites present diverse chemical structures. Over 2 million secondary metabolites are known, but although the high diversity, they are originated from a restricted number of metabolic pathways. This metabolic pathway is the metabolism is grouped in 3 classes: terpenes, alkaloids, and the phenylpropanoids. These compounds are identified in all plant tissues, for example, in leaf, root, or seeds, and their formation and gene regulation, where they are going to be produced and when they are going to be produced is regulated, usually organ, tissue, cell, and also development specific, indicating that many transcription factors must cooperate to transcribe secondary metabolism genes. So to produce a secondary metabolite, the genes involved must be on to in a specific plant organ and in a specific plant development. Many secondary metabolites are source of drugs. However, chemical synthesis is not economically viable, and isolation for plants still represents the only option. Another option is the bioproduction. So to do that, we need to apply biotechnology techniques such as transcriptome, that you identify the genes involved in the biosynthetic pathways. And once you identify these genes, it's possible to replicate the biosynthetic pathway in an organism, in a model organism, to produce secondary the metabolites in a model organism, such as Saccharomyces cerevisiae. But to do that, to identify the enzyme involved in metabolic pathway and the gene sequence of these enzymes, we need to apply to perform a transcriptome profile. So it's important to know the enzymes involved in each metabolic pathway and the corresponding gene sequence of these enzymes to reproduce this metabolic pathway in another organism. For example, Maithanus elicifolia is a Brazilian native plant from Celastraceae family, and this plant is a tree, and this plant is known for its variety of therapeutic properties. For example, Maithanus elicifolia leaves and the root has been used for a long time as a treatment of several diseases. Some compounds are synthesized in specific tissues. For example, canonomethydius triterpenoids are accumulated in root, and flavonoids are found in leaves. So the core of this project is to analyze, to proceed with an RNA sequence to analyze the differentially expressed transcripts between leaf and root to provide a better understanding of genes involved in secondary metabolic pathways. So we used the Maithanus elicifolia to perform this analysis, and we produced the whole transcriptome of Maithanus elicifolia and identified genes involved in biosynthesis of secondary metabolites by comparing the profiling from root and leaf. So we wanted to know the gene sequence of the enzymes involved in secondary metabolic pathways using this tree, Maithanus elicifolia, to identify genes involved in metabolic pathway of secondary metabolites. To do that, first, the total RNA from leaves and roots samples was isolated and the cDNA libraries were prepared. These libraries were submitted to the next generation sequence, and after sequencing, the raw data was assembled using Trinity. Trinity is a RNA-seq de novo transcriptome assembly software. Other softwares were used to produce more results, and I'm talking about doing the results, the next part of my presentation. So, talking about some aspects of the transcriptome, the whole transcriptome, the first thing that we need to do is to make an union from transcriptome from leaf and root, and we found more than 163,000 transcript with AGC content is a trait of the transcriptome, about 41%. This value is similar to those reported for other species from the same family, Celastraceae. Consider transcriptomes identified in leaf and root individually. The majority isoforms were found in both tissues. Okay, so the whole transcriptome, all transcripts in the plant, is coincident in both leaf and root. And the individually transcriptome are similar about the traits of the transcriptome. For example, GC content, the median length, the average length, they are similar. Another aspect that we can talking about the transcriptome is comparing the transcriptome with other transcriptomes that are in the literature. So compare the transcriptome sequence to another organisms using BLAST-X. The Methanus alexifolia transcripts have highest similarity with organisms classified in the same family, Celastraceae, and mainly it's very similar to Pterigium wildorf. This is a species very close to Methanus alexifolia in terms of taxonomic. After application of some filters in the transcriptome, for example, we had this number of total of transcripts. After we apply a filter to low expression, and we found this number of transcripts, and so on. We found more than 2,000 of the transcripts are involved in metabolic pathway. So we had initially this number, and finally we need to look to this number of transcripts to find transcripts involved in metabolic process. And if we looking for the comparative transcriptome, looking for which genes are mainly expressed in root or mainly expressed in leaf, we can find this number of genes mainly expressed in root, and this number of genes mainly expressed in leaf. It's not exclusively, it's mainly expressed. And we can find this number of enzymes involved in the transcriptome that are that have a difference in expressions. So we have 350 that are enzymes that are mainly produced in root, and this number that are enzymes mainly produced in leaf. So we have a good filter to find enzymes involved in metabolic pathway, in secondary metabolic pathway. For example, in root, when we are looking for genes involved in secondary metabolism, looking for these numbers of enzymes, we mainly find this kind of biosynthetic pathways. Enzymes involved in monoterpenoid biosynthesis and isoflavonoid biosynthesis were identified in root, while flavonoid biosynthesis and biosynthetics of alkaloid derived from histidine and purine in leaf. So finally, from the present study, an extensive transcriptome dataset has been generated from the de novo sequence analysis of Maitenus ilicifolia, the first work that of transcriptome analysis in Maitenus ilicifolia. This work now can coverage of the transcriptome data consistent to discover genes involved in the secondary metabolic pathway. So we have now more work to do to confirm that these genes are really involved in these metabolic pathways that we are interested for. So that's it. Thank you very much.
Okay, so we had our third presentation in a record, and so we're gonna present the last presentation in this session, a short paper presentation entitled Math Pipe Classification of Pro-Inflammatory Peptides Using Mathematical Descriptors. It will be presented by João Pedro Cavalcante from USP São Carlos. You're live.
Yeah, I'm here.
Oh, nice. I was about to announce your record, but now we can have you live. Okay, João Pedro Cavalcante from USP São Carlos will present to us live. You have 10 minutes sharp,
Okay.
and then we're gonna proceed for the questions, not only questions about your work, but also about concerning the previous presentations.
Okay, I'll share my screen now. Just a second. Can you see?
Yep. You're sharing. Okay.
Okay, is full screen okay?
Yes, it's nice.
Okay, can I begin?
Yes.
Okay. Hello. As he said, my name is João Pedro. I'm here to present you the MathPip classification of pro-inflammatory peptides using mathematical descriptors. I'd like to thank Anderson, Robson, Danilo, and André. They are my co-authors for this opportunity, and BSB also. So I'm going right in. First, I'd like to talk about what we are tackling. What is the problem that we suggested? First, it's a machine learning classification of biological sequences. By biological sequence, I mean any material or object that has DNA, RNA, or something that's a sequence based on AGCTA, the classic biological basis. So there is a lot of biological sequence available out there by research teams by every place in the world. So it's very important for us to harness this potential, even though we didn't do a lot of research on it before. And that's the data that we want to tackle, data that has not been already researched, because it takes time and money to study it. And in practice, we want to harness the data that's out there. So the data that we chose to work this problem on was pro-inflammatory peptides. That's a type of peptides that do part in our inflammatory response on our systems. It's a biological sequence. As I said, it's important for us. It's relevant because it's one of our primary defense mechanisms of our body. So it's well documented, and it's important right now because of COVID-19. That's a part of the infection of the disease. And also, there are previously studies that have used this data set, so we can have a basis for comparison, which is very good for us. So what's in the literature right now? The use of machine learning for classification. That's what's in the literature right now. But first of all, machine learning is a program that learns inputs and outputs to get inputs and produce new outputs that classify things. Right now, people use a feature engineering that is create information based on prior knowledge of the biological sequences. So if it's let's say a protein, people would get physiochemical properties of this protein, and they would try to create physiochemical properties. Like there is this, for example, a starting codon would be prior biological knowledge, things that we already know because we've studied. But the problem is our data, what we want to harness is data that has not already been studied. So we don't know anything about it. And thinking about this, we came up with a new approach that's called intermediary representation for short. It's also a machine learning based, but feature engineering. But the difference is we translate the sequences into an intermediary representation. And from this intermediary representation, we extract features, informations, and this kind of thing. The sequence to an intermediary representation, and the intermediary representation, we get the feature itself. It's mostly math-based, what we do. So what I mean by that, this is one example of an intermediary representation. This is the VOS transformation or VOS representation. It's basically a rule of translating biological sequences into number-based sequences, discrete number sequences. As you can see, if it's an A, it will become an array of 1, 0, 0, 0. If it's a C, it will become an array of 0, 1, 0, 0, and so on. On the bottom, you can see a simple representation of a translation of a simple sequence. So this is the intermediary state. But how does the feature engineering goes? Since we translated our sequence to an intermediary number-based representation, we can treat it like a function, in essence, a discrete function at least. So here we represent it on VOS, and now we are using it as we are extracting the features as with Fourier transform. In this image, you can see audio being translated from time to frequency, but we can translate the sequence itself into a number, and this number and this discrete VOS number, we can translate it to a spectrum. And we can analyze the spectrum to differentiate between sequences. And that's what our machine learning code will be receiving. For these transformations, this was one example. VOS is just one example, not everything that we used. Math feature was used. Math feature is a tool that was published recently on other journals. It was developed by Hobson, one of the co-authors, and it helped us a lot in the process. So now I'm going to the experimental setting. There are metrics. Our metrics of machine learning models were accuracy, Matthews correlation coefficient, and AUC. The first two ones were for purpose of comparing them to other papers. And the last one was we measure how the model was going. We used the benchmark dataset, so it's the same dataset as other three works. Also, the way that we studied the metrics, there were two ways. The first one was the cross-validation tenfold. That's basically It's normal for any machine learning project to use it. And the train test split. The train test split, we did it the same way that the other works did. So we could have a basis for comparison using the same rules. These are the results. They are quite good on the cross-validation, and they get a little worse on the train test. What's to be expected, since cross-validation is a mean between a lot of iterations of the same data set. And the final results, we were better than 2 of the previous works, a lot better, but worse than the Pro-Infuse. The explanation for this is probably because Pro-Infuse used more sophisticated machine learning methods to develop and we did not. We just wanted to check how well would our generic math-based representation of features work out against normal informations. And I think that based on these results, we can say at least that it's good to take a more deeper look into what we proposed. It's important. So about the references, these are the references of everything that I presented. Other references like the COVID and things that I cited are on the actual paper. That's the end of the presentation.
Thank you so much, João Pedro.
Oh, thank you.
So now we move to the second part of our session, in which we address questions to the speakers. So we have here João Pedro, Fernanda, and Pamela. So let's take a look at the chat, YouTube chat. A question about the paper. Record no. No one, no questions. Okay, so I'll address you some questions. I will start with Fernanda. In your work, I found very interesting that EIF5A is the I researched on the Wikipedia, and I saw that it's the unique human protein that's known to be hypogenated. So it's that the uncommon amino acid is present into a single protein in the human protein. I don't know if there are more proteins in gist that has that modification.
No, it's exclusive. Just EIF5A has a hypogenation, and we have an ortholog in prokaryotes that it's EFP, but it's the only protein that undergoes hypogenation. It's an exclusive amino acid.
And it seems to be essential for the metabolic balance in the cell.
Yes.
And I was wondering if you checked autophagy, it's because you're looking the side effects of that mutant protein, which is impairing the whole metabolic within the cell. And I was wondering whether it would be interesting to carry out analysis such as flex balance analysis, FBA, and the dynamic modeling like that to further understand the impact of the absence of hypogenation in the cell's metabolism.
Yeah, well, we have in our lab lots of mutation. We have 3 mutants from EIF5A. We have this 11 mutant that it's only that doesn't hyposinate, right? And we have two other mutants that works on that are doesn't have not doesn't have because it wouldn't be viable, but has a decrease in EIF5A levels. But I worked with these other two mutants, and the results were not so, let's say, interesting as with hyposination. So one of the possibilities that we came with my work, but we didn't got to an end of it, is that actually hyposination only occurs in the cell to work as a, let's see, to get rid of polyamines in excess. Because spermidine is hyposination is spermidine dependent, and high levels of polyamines are also toxic to the cell. This was one of the hypotheses that we came to with our study, but I can't say that, right? I can't write that because we don't have more experiments to prove that. But it's really hard to work with EIF5A because of these situations that if you how can I say, if you exaggerate in the amount of EIF5A that you are getting rid of, it doesn't grow up, right? So that's the problem. But I have a friend of mine that is working with modeling and EIF5A. She is searching for drugs for malaria and these diseases that are, what's the name?
Like a tropical disease.
Yeah, not when we don't care about the disease.
Neglected.
Yes, that's it. That's the word. So she's working with modeling to maybe find a drug to fight neglected diseases.
Okay, thank you. Now I would address a question to Pamela. Pamela, your methodology to evaluate the primer's efficiency, it is very relevant, very important, especially it's a relief to know that those primers are very efficient to detect variants of concern. And the applied methodology itself is there's a novelty in respect to what other people, other groups do to assess primers efficiency. What's the main contribution of the methodology, applied methodology?
Because we use the recently parameter sets I mentioned about the DNA mismatch. It was evaluated if it was parameterized from an experimental set of melt temperatures for single, double, and triple mismatches.
Okay.
So it had many combinations. It was about 4,032 experimental sequence with mismatches. So it was about more than 4,000 combinations. So it is, how can I say, little more accurate in comparison to other methodologies we tried. So we combined a statistical physics model with these recently parameters. So we believe it is a little more close to the behavior of them, of mismatch.
Okay. And could you predict from that you gather a lot of experimental data? Could you predict, for example, if I give you a new variant of concern, for instance, lambda one, could I use your methodology to infer the the primer's efficiency for that new
Yes.
variant of concern. Yeah,
Yeah, we are
that's
already designing the primers for oncogenes, so the model could be from the melting temperatures and vice versa. So you can do both ways.
okay. Okay, thank you.
Thank you.
Now I'll address that question for Pedro, João Pedro.
Okay.
João Pedro, I I found very interesting that you're using Fourier for to extract the features from
Mhm.
your sequence data. Myself, yeah, I'm interested in that problem, but in using it to extract the descriptors for time series, for instance, temporal measurements of
Mhm.
proteins instead of sequences. But nevertheless, in your case, you showed that you could you could get good results using those descriptors to train a machine learning model.
Yeah,
However, and it's another nice thing that you showed that it's not beating the perhaps the
the
current
best one.
gold standard, the best one,
Yeah,
current gold standard, which is Pro Infuse.
Pro Infuse. Yes.
But there is a it is important, and you can tell me if you agree with me or not. It would be very important to try to get the Pro-Infuse and feed it with your mathematical description and see if you can beat it.
Yeah,
The wild type Pro-Infuse against the Pro-Infuse that is adapted to mathematical descriptions, because
yeah.
the core of your work, as I could, if I understood correctly, is
Mhm.
the descriptor, not
Yeah,
the machine learning method itself.
yeah. Not the machine learning itself, but the thing is, we have a machine learning model. So this model is trained with data, and the kind of data that this model is trained will also be what will define what the parameters of the model will be. So if I have a gold standard model, I also have a gold standard feature engineering process, and you can change that feature engineering process with the without changing the model itself. So I can't just we can't just feed a model that has a different feature engineering um, process with other features, other information. What could be done is try to use the same techniques. So they used a technique called ensemble that's they use they unite some models to make a prediction. It's like a voting based system of many models, and we could theoretically replicate that, but the model itself from Prometheus can't be used with other our feature engineering or any other by this case, because that's a limitation of how machine learning models work.
Okay, so you say the whole if you try to change the how the model is fed,
Yeah.
actually you use a new model, not it's not the
Yes.
same.
That's
Okay,
exactly. Yeah. That's it.
right.
Yeah. There is a saying in data science or machine learning that your model is only as good as your data, and you have to present your data in various ways. So if you change your data in practice, you're changing the whole model itself.
Now, I have a second question based on that,
Hmm.
which is why if get you applying a mathematical descriptor to extract features
Hmm.
using a technique like the Fourier transform.
Hmm.
Why you get features that are more suitable for machine learning. Instead, use the whole set of raw data directly.
Well, the reason why I can't really tell you, justify, it's all we can do is test the model itself with a representation. So if the representation is a number, if the representation, let me give a simple example. Imagine we are trying to do a regression on a store, like the price regression of shoes, let's say. And the shoes have sizes. One way that I can represent my data, that is the shoes, with the size, I can do a size that's a medium, big, small shoes, like three words. Some models can take that and use it to classify things or to regress in this case. But if I call my shoes 1, 2, 3 size instead of small, medium, big, some models will yield a better result than if I call them small, medium, and big. So the way that we encrypt the data be the sequence, getting information from it, from biological known things from the sequence, or doing a math-based extraction, what I do, can only be as good as the model can make it. Some models wouldn't be able to get this data and work with it. Other models would. I don't. I think if I
Yeah,
didn't respond it well, you can tell me.
no, you responded. You answered well. So you are saying that instead you are working with categorical variables.
Yeah.
You're using labels in which you have ordering.
Yeah.
You are putting more information. This
Yeah.
the Otherwise you wouldn't put more in gain information to improve your machine learning. So perhaps with your transformation you are putting information that otherwise wouldn't be there.
Wouldn't be there. Yeah. Yeah. I'm trying to extrapolate information that's not really present just on the sequence alone. But the thing is, no, that's it. Okay.
Okay. Um, that's for Pamela. It's not exactly a question, but just a remark. Your work has also a software. You developed a software.
Yes, we developed the pipeline, the workflow, so we available all the programs, we have the documentation if someone wants to do on my own. So there is all the scripts and the examples because we apply it to other parameter sets. So there are all the primer sets for PCR and LAMP also.
Okay. Uh, okay. Thank you. I have. I think I have a question here on your YouTube. It's a question from Professor Raquel for João Pedro.
Okay.
It was not clear which peptide database was used. You mentioned that nothing was known about peptides, but I understand that the base is labeled in inflammatory and non-inflammatory peptides, right?
Mhm.
Or
Yeah.
misunderstood?
Yeah,
Is
you're right. Oh, sorry.
so
Okay.
just to complete, there is an inflammatory peptide benchmark, right?
Yeah. So I think in my hurry to present, I did the confusion. Sorry. Um, First of all, the data that we would like to ideally in the ideal world that we would like to um, be able to utilize is data that is just a sequence, and that's it. We just have the sequence of the object, nothing more, nothing that we know. That's the theoretical problem. But we need data that's already labeled and already known, and we can compare to other works. So we use the peptide, pro-inflammatory peptide base, benchmark base, that other three studies also used. You can find it in the ProInfuse study. That's also quoted in my study, so in my short paper. And I don't remember right now the link. I think it's iedb.org. I don't remember where you can get right now. But if you look at my paper, you can go to the ProInfuse paper, and the dataset itself will be there. It's a benchmark dataset. I think it is
Yes,
the
you answered. Actually, I saw your short paper, but I overlooked that you you didn't mention in the presentation itself. But uh, and just to recall who attended yesterday the machine learning introduction mini course, always a question whether you are using labeled or unlabeled
yeah,
that data. It's relevant
supervised
to
and unsupervised learning. That's
yes. Okay, I don't think we have more questions from the attendance. So I'd like to thank you very much, you three, for your talks and your nice talks. Congratulations to you all. And for the fourth presentation, the Mariana's presentation, which was recorded, the audience can send questions to and we from the organization we deliver to her later. And I'd like to make at the end of this session a quick announcement. After lunch at 2 p.m., we're gonna have our second mini course. It will be presented by Dr. Luciana Santos. It's a docking mini course, very interesting subject. And I I mean, I ask everyone that's interested to participate in that in that mini course. It will be again in at Zoom. You need to register yourself in Zoom prior before 2 p.m. in order to have your pre-approval and guarantee your access to the mini course. So thank you again for our speakers. Thank you the audience for joining us in this first technical session. See you in the evening, in the afternoon, 2 p.m. for the docking mini course. Thank you.
Thank you.
goodbye.
Bye.
Thank you.